Hello, everybody. My name is Reilly Mach, and I am a student at Concordia College in Moorhead, Minnesota. And for my presentation, I'm going to be talking about S-nitrosylation of palmitoyl protein thioesterase 1 in neurodegenerative diseases. This research was performed at Scripps Research in La Jolla, California, under the mentorship of Tomohiro Nakamura and Stuart Lipton. So to start off with some background on S-nitrosylation, physiological nitric oxide signaling contributes to the maintenance of normal neuronal activity and survival, and this serves as an important regulatory mechanism in the central nervous system. However, exposure to environmental toxins or the normal aging process can trigger excessive production of reactive nitrogen and reactive oxygen species, such as nitric oxide, and this contributes to the etiology of several neurodegenerative diseases. So the S-nitrosylation specifically is an important effector of nitric oxide signaling in both health and disease conditions. To show you a schematic of the S-nitrosylation reaction, what we see here is that it's a redox-dependent post-translational modification, and it features the covalent attachment of a nitric oxide group to a cysteine thiol of a target protein. So here we have the cysteine, and then we attach a nitric oxide group to that thiol. So thinking again here, really the key is that elevated nitric oxide levels might represent this molecular key behind diseases such as Alzheimer's, Parkinson's, ALS, etc. And these elevated nitric oxide levels lead to an increase in S-nitrosylated proteins, and S-nitrosylation of proteins can lead to protein misfolding and a reticulum stress and mitochondrial impairment, all which are not ideal conditions for physiological well-being. So here there are potential pharmacological therapeutic approaches to help mediate this neurodegeneration. So in this study, human brain tissues were analyzed and S-nitrosylated proteins were captured. So there were 58 S-nitrosylated proteins found exclusively in the hand, which is HIV-associated neurocognitive disorder, plus methamphetamine use. So here, these are these 86 proteins here. And this group is where PPT1 was identified. And these identified proteins are found functionally involved in metabolism, immunity, and neuroinflammation. And essentially here, what we see is that the identification of PPT1 within this study suggests that it may be somehow involved in neurocognitive diseases. And additionally, with meth use, we see a kind of double whammy there of disease conditions. And so the fact that PPT1 was identified here kind of signaled that it was a protein that we should look more into and try and understand further. So importantly, similar to S-nitrosylation is the process of palmitylation and depalmitylation. So S-palmitylation is the post-translational reversible covalent linkage of a 16-carbon fatty acid to proteins, and it increases the protein's hydrophobicity, and that facilitates interaction with lipid membranes. It also acts as a post-translational switch, similar to phosphorylation here. And palmitylated proteins require depalmitylation, which is the removal of palmitic acid prior to lysosomal degradation. This demonstrates the significance of the process in protein sorting and turnover. If it's going wrong, we see a lot of protein aggregation seen in neurodegenerative diseases. And both of these processes, palmitylation and depalmitylation, are important regulators of protein activity in synaptic function and neuronal development. And there are three classes of enzymes that facilitate this depalmitylation reaction, which are the acyl protein thioesterases, alpha beta hydrolase domain containing 17 proteins or ABHD17s, and the palmitoyl protein thioesterases, which is where PPT1 is involved here. So just a schematic to show you the palmitylation and depalmitylation reaction. This red circle is where PPT1 is identified, as it will chop off this carbon chain, leaving the cysteine thiol unmodified. So some key features of PPT1. Like I've mentioned, PPT1 is palmitoyl protein thioesterase 1, and it is a depalmitylating enzyme, meaning that it removes that thioester-linked fatty acyl groups like palmitate from cysteine residues, and this is again important during lysosomal degradation. Loss of function mutations really can cause devastating diseases and often can lead to those neurodegenerative diseases. And S-nitrosylation may affect this activity of the enzyme, which is one of the aspects that I looked into during this research project. And importantly, going back a little, PPT1 is the first depalmitylating enzyme to be linked to a genetic disorder, which is also what identified it as a target protein that we may want to look more into. The hypothesis here of this research is that dysfunction of PPT1 may be implicated not only in pediatric neurological diseases, but also age-related neurodegenerative disorders. So to go into the results of the project, what I was actually able to find throughout my summer at Scripps Research, really what we can see is the first thing is PPT1 is localized in neurons. And so this staining was done in rat primary cortical neurons. And here we see it. We have red is the MAP2 neuron stain, the green PPT1 stain here, and then Hook's is just a neuronal stain. And so importantly, what I want to show here is the MAP2 plus PPT1. We can see a lot of overlap between these red neurons and the green PPT1, which is super interesting. And also really, really interesting here is some of these dots within the PPT1 stains. If you can see, that's actual localization to the lysosome, which is really interesting since we know PPT1 is involved in lysosomal degradation. It's very cool to see it represented there. Not only is PPT1 localized in neurons, but it's also localized in glia. So GFAP is an astrocyte stain here. So we see GFAP, these are red, astrocytes, PPT1 in green again, and the overlap, I have an arrow kind of representing the astrocyte as well as the PPT localization, which you can see throughout. And then IVA1 in green here is a microglia stain. And then in this one, PPT1 is red. So these arrows in the combination represent that overlap where we can see PPT1 protein also overlap with the microglia, and that shows that localization there. The crystal structure of PPT1 demonstrates that we have a disulfide bond here between cysteine 152 and 160. And these yellow means that they were identified from proteomic data. The red here is other cysteine residues not found in the proteomic experiment, but we do know that cysteines are there. Magenta is catalytic residues required for enzymatic activity to occur. And in this case, PPT1, these would be required for depalmitylation. And then palmitate is here in blue, and that's the carbon fatty acid chain. Another important finding here was that before we were able to do the experiment, we needed to know whether or not there were even free cysteines to target. In order to do so, we did a mobility shift assay, adding pegmalamide to the sulfurs here. And what we were looking for, sort of in the schematic, you can see that here's the unmodified protein structure. And then when we add pegmalamide, we can see that mobility shift to a heavier protein because pegmalamide should cause about a 5 kilodalton mobility shift. And that signifies that there are sulfurs to target since pegmalamide would have been able to bind. And we can see that here, the results demonstrated exactly what we were hoping. So we knew there were free cysteines to target. Going on, knowing that there were cysteines, we decided to go on with the experiment, and we wanted to see whether or not S-nitrosylation was occurring in PPT1. And we did this, we found this through a biotin switch assay, which is a biochemical assay using Western blotting to detect S-nitrosylation of target proteins. Specifically here, we were looking at PPT1. And so you can see we have a control, we have SNOC, which is a physiological nitric oxide donor. And then we have SNOC without ascorbate, which is a positive control there, negative control, excuse me. And so the fact that we can see S-nitrosylation band here demonstrates that PPT1 is capable of being S-nitrosylated after rat cortical neurons were exposed to SNOC. So we did find in these rat cortical neurons that PPT1 is S-nitrosylated, which signifies that it is somehow involved in the neurodegenerative diseases. We also confirmed these findings in human HEK293 cells, which is really interesting. And again, we can see similarly in our SNOT category here, you can see the signal suggesting that PPT1 is S-nitrosylated. And in the human cell line, we actually repeated the experiment enough to generate statistical significance between the control in SNOC and the SNOC versus ascorbate control groups as well. And so really this is interesting because we can see that S-nitrosylation occurs in 2 different cell lines. So the main findings here, first confirmed localization of PPT1 to neurons, astrocytes, and microglia. confirmed a presence of free cysteine in PPT1 through the mobility shift assay, confirmed that PPT1 is S-nitrosylated in both human HEK cell lines and rat cortical neurons. Some ongoing and future plans. First, we'd like to examine if S-nitrosylation affects PPT1 activity. We'd also like to continue mutagenesis experiments to identify the specific cysteine site of S-nitrosylation. And finally, confirmed that endogenously produced S-nitrosylated PPT1 in animal models and post-mortem human brains to see whether or not this occurs without that SNOC physiological donor. So some of these ongoing projects were started in the last couple of weeks of my summer at Scripps, and so I'll tell a little bit of these preliminary findings, but none of this is conclusive due to it just being done 1 or 2 times. So with the enzymatic activity assay, we use a fluorogenic substrate, which measures fluorescence increase after the substrate is cleaved from PPT1. RFU here is a relative fluorescence unit, and that's a signal measure. We overexpressed PPT1, but when we did it, we realized that we probably overexpressed PPT1 a little bit too much, and we want to use a little bit less of that in the future if we were to repeat this experiment. But we do see a slight decrease in enzymatic activity following SNOC treatment, but we don't know for sure whether that's true. In order to see a more significant decrease, we would need to use less PPT1 expression in order to kind of quantify that more significantly. And we also propose that changing the incubation time during SNOC treatment may help generate more conclusive findings here. I was also able to do some mutagenesis experiments and see where exactly the specific site of S-nitrosylation is, which cysteine. And so we produced 12 mutant plasmids here. Each of these represents a different cysteine to alanine mutation. Some of them are double mutations, or some of them are just single, such as the C128A here. And then we also added a Mach epitope tag on some of these as well. So the double band on the input here may be due to mutation, could be caused by some sort of conformational change. And it's likely due to now being able to form a disulfide bond between the cysteine 152 and 160, or being unable to form that bond since we changed both of these to alanines. We no longer see that cysteine disulfide bond there. And when we look at the C128A mutation, this may have increased signal due to more free cysteine 96s. If we look at the map here, the 128 and 96 are relatively close and may have had some sort of secondary structure there, tertiary structure. And so if we mutate this one to an alanine, we might have had more free cysteine 96, which could suggest that this might be involved in the S-nitrosylation reaction somehow. So that is all for my presentation today. I hope you all got an understanding of the S-nitrosylation reaction and what that looks like. It's very interesting research summer, and I'd like to offer thanks to the Lipton lab members at Scripps Research. I had a great experience, and I'm so glad to be able to present this research at the Sigma Zeta convention today with you all. And I hope you enjoyed the presentation, and please reach out to me with any questions if you have them. But thank you so much.